Infi, can you tell me how to design an organism that will help with climate change? Welcome to CognitionChunks. This information is for entertainment only. I am not trained in genetics. Is this useful? Is this nonsense? Join me as I attempt to learn from GPT. Here is Designing PhytoSynthetica.
Inventing a new, useful species involves a multidisciplinary approach that encompasses genetic engineering, synthetic biology, evolutionary biology, and ecology, among other fields. The design of a new species must consider not only the genetic and biological aspects but also ethical, environmental, and practical considerations. For the purpose of this exercise, let's conceptualize a beneficial species that could contribute to environmental sustainability and human well-being, which we'll call PhytoSynthetica. Step 1. Conceptualization and goal setting. Objective. Create a species designed to combat climate change by efficiently absorbing atmospheric CO2 and converting it into oxygen and biomass, with the added capability of producing a biodegradable material beneficial for human use. Step 2. Genetic programming for novel proteins. 2.1 Enhanced photosynthesis. Gene design. Modify the ribosio enzyme ribulose 1,5-bisphosphate carboxylase/oxygenase, which is responsible for CO2 fixation in the Calvin cycle but notoriously inefficient. Engineer a version with a higher affinity for CO2 and reduced photorespiration rates. Protein engineering. Introduce genetic sequences from extremophile organisms that possess versions of ribosio with these desired traits, possibly integrating sequences from chemoautotrophic bacteria that exhibit high efficiency in carbon fixation. 2.2 Biomass conversion. Gene design. Integrate genes responsible for the production of cellulases and other enzymes that break down plant biomass into simpler sugars. These genes could be sourced from organisms like Trichoderma rhizae, known for its potent cellulase production. Synthetic pathways. Create a novel metabolic pathway allowing the conversion of fixed carbon into a biodegradable polymer, similar to PHA polyhydroxyalkanoates, but with enhanced properties such as increased durability and flexibility. 2.3 Environmental adaptation. Gene design. Incorporate genes for desiccation resistance, salt tolerance, and temperature resilience from extremophiles, enabling the species to thrive in various climates and reduce competition with native species. Protective proteins. Introduce antifreeze proteins from Arctic fish and heat shock proteins from thermophilic bacteria to provide a wide range of temperature tolerance. Step 3. Cellular architecture. Chloroplast modifications. Engineer chloroplasts with increased surface area for light absorption and integrate artificial antenna complexes that can capture a broader spectrum of light, enhancing photosynthetic efficiency. Energy distribution. Design a novel organelle or modify existing ones, e.g., mitochondria, to efficiently channel the energy derived from photosynthesis towards biomass conversion and polymer synthesis. Waste management. Implement a cellular mechanism for the safe breakdown and expulsion of any toxic byproducts resulting from synthetic processes, ensuring environmental compatibility. Step 4. Organism design and development. Morphology. Design the organism to have a fast growth root system for carbon sequestration and a leaf structure optimized for light absorption. Consider a modular design that allows for easy harvesting of the biodegradable material it produces. Reproduction control. Engineer a fail-safe mechanism to control reproduction, possibly through a synthetic auxin system that can be activated or deactivated externally to prevent uncontrolled spread. Step 5. Environmental and ethical considerations. Ecological impact assessment. Conduct comprehensive studies to understand the potential impact on local ecosystems, ensuring the species does not become invasive or disrupt native populations. Regulatory compliance. Work within the framework of genetic modification regulations and biosafety protocols to ensure ethical development and deployment. Step 6. Implementation and monitoring. Pilot programs. Introduce the species into controlled environments to monitor its growth, ecological impact, and effectiveness in carbon sequestration and material production. Feedback loop. Use data from pilot programs to refine the species design, addressing any unforeseen issues and optimizing its beneficial traits. This step-by-step approach to designing PhytoSynthetica illustrates the complexity and interdisciplinary nature of creating a new, useful species. It's essential to proceed with caution, ensuring that such innovations are beneficial to both humanity and the environment. This has been an episode of CognitionChunks. Join us for the rest of this series as we explore this topic in more detail. Please like and subscribe.